I'm sure you're all very anxious about the final exam. The official exam is May 13th on a Monday in the morning at Harvard Hall. Actually, where is Harvard Hall? You guys know?
Yeah, it's like sort of at the entrance of Harvard in the yard.
Oh,
like,
okay. So you guys all know.
Yeah, it's like
I see. Okay, so it's Harvard Hall 201. We'll have people proctoring exam there. The alternative exam time is only for graduate students, and you have to tell me ahead of time. Okay, otherwise, undergraduate student, you need to get special permission from the registrars if you want to take alternative exam. It's Tuesday afternoon in the Longwood campus. This is open notes. You can look at your notes. It's only open computer because we don't want you to print out all the slides. We do not want you to search for, oh, how do I do this? Please do not do internet, search or chat with your friends about this question. Just make sure this is standard honor code. Yeah, so it's open computer only in that you can look at what you already have on your computer. Your slides, your homework, whatever you stored. We just don't want you to print out like stack of PowerPoints. Okay, that's all. Other matters, yeah, make sure you watch the last lecture video on reproducible research. We would appreciate you to fill out. So each homework still have the very short survey, but we hope you can fill out the course evaluation. Previously, we mentioned there's extra credit for videos. If you can find the videos already better than what we have included in the course website, we include some other people's video, please let me know, especially on the newest topics, because some of these new things, they are not very good videos that thousands of people have looked at. But to make sure that, first of all, this video needs to be short. I think in general, it's very hard for people to watch a 1 hour, 15 minutes video from something, right? So usually, if it's like 10 minutes, 20 minutes, you can learn some topic. That's great. Make sure it's very clear. I think an example would be like StatQuest, like from earlier this semester. You can see those kind of the lecture videos we hope to include, and really to the point, very clear on a very specific topic. If you can find those, please directly email me. And if I do include that in next year's class, I will give you some extra credit. Make sure not only you include that, you tell me what topic it is on, why it is better than what we have, okay? So, and another one, this will come in the form of a survey. Well, actually, you know what? We will create another survey. How about this? We will include all of them as a survey. This is one survey, this is another survey. But improve the homework questions, we've seen that, for us, actually, we're trying, I hope you can appreciate now, with each homework, we try to use one data type to kind of show you, oh, this is how you look at like glioblastoma. This is how you look at ChIP-seq of some prostate cancer factors, right? So for that, we try to fit all the questions together. Very often, we have to create this question and run this ahead of time because original study may not have done everything together. And so very often, we finish running the tools to make sure we get the result as we wanted, like the day before we give it to you. And so some of them, it could be that, shoot, this result is not so good. Let's try a different data set. And so as a result, I think the questions may not be in the most, as some students say, oh, it took me a while to figure out what this question was really asking. So in the survey, we're going to ask you which homework, which part, and which question. That question could be rephrased into something that you think can improve the homework for future years of students. Of course, we might also change the data set in the future, but at least I can see where the potential problems are. Okay, so let me give you some example questions. We will have some true or false questions. Let's try it. For microarray experiment, we should use p-value less than 0.05 to call differentially expressed genes. What's the correct answer? Yeah, if you need to do multiple hypothesis correction, yeah, probably FDR 0.05. Multidimensional scaling can perfectly capture the pairwise distance between nodes. No, because it will try to approximate, but sometimes there might be a little conflict. The more dimension you allow it to have, the more likely it will approximate the original pairwise distance. BAM file is usually used to store the coordinates of genomic intervals. False. Yeah, BAD file is really for this. BAM file is for read mapping, right? You get a BAM file after you map the reads. Epigenetic information is encoded in the DNA sequence. Yeah, epigenetic information is inherited but not encoded in the DNA sequence. A gene can be an oncogene in some cancers, but a tumor suppressor in other cancers. That's true. Okay, so you get some idea, true or false. It should be like really fast. You should understand this quickly. Okay, multiple choice. Which of the following linkage considers pairwise distance of nodes in hierarchical clustering? Average linkage. Compared to SMART-seq, droplet-based single-cell RNA-seq method has. It has 3' bias. They have UMI. They are not more accurate per cell, but they are kind of more accurate when you look at a cluster. Right? So it's a B and C is the answer for this. So you get some idea. Trust me, I will not ask you the same question if you have already asked it here. Okay, so we will ask you some short answer questions. Let's say, if you, these are two different questions. If you use hidden Markov model to predict a protein's secondary structure from amino acid sequence, what are the parameters in this hidden Markov model? And what do they mean in this scenario?
The hidden state is the secondary structure.
Yeah, the hidden states are the secondary structure. What's your observation? Is the amino acid at each location, right? What are the parameters? Yeah, you have transition from
secondary.
Yeah, different like alpha helix, beta strand, loop, right? These are the states, and then the transition probability. What's the initial probability? Yeah, right. You can probably just look at the current protein database. With the structure, you ask how often does the protein start with alpha, beta, or loop, right? And what is the emission probability in this case?
Given the secondary structure.
Yeah, given the secondary structure, what's the likelihood you are gonna see a valine or a lysine or some whatever amino acid? And you can also use the current protein database to say, okay, in the existing structures, how often do we see a valine or a lysine or whatever in alpha helix versus beta strand, right? So you can estimate that. Okay, now second one is it's about multiple hypothesis or whatever hypothesis testing in general. Hypothesis testing in general. We are asking, these two group genes are differentially expressed or not differentially expressed, versus whether you call it as, in your seq or whatever, call it as differentially expressed or not. What is the sensitivity? So sensitivity is if this thing is indeed differentially expressed, how often you call it, right? It should be, let me see. I think it's S divided by this, right? So the two groups indeed are differentially expressed. How often am I calling it correctly, right?
So this is number rejected.
Yeah. When you reject a null hypothesis, it means you call it as differentially expressed, right? What is the true negative rate? So true negative is they are not differentially expressed, and how often do you not call it? It should be U divided by M0, right? What is the false discovery rate? What's the false discount? Yeah, it's V over R, right? So among all the things you call it as differential expression, which ones are, or what percentage are wrong? Positive predicted value, which is if you call it as differentially expressed, they are indeed differentially expressed. It's S over R. Sensitivity is, if they are differentially expressed, it's S over M. It's S over M1, whereas positive predicted value is you call it as different, whether it is indeed different. Right, so you can get some idea. Right, so that's kind of another example of a short answer. Okay, now long answers. Actually, this is the key. I'm hoping that after this course, if some people come to you for collaboration, you can roughly see, okay, you tell me, or you just say, okay, what experiment have you done? What biological questions are you trying to answer? Do you have some hypothesis, right? And we say, okay, think about this. You can say, okay, you know, this data, I'm going to do A, B, C, D. And if I get this, I will do this. If I get that, I'll do this. And also you can say, well, if I get this result, maybe this group should do this, whatever experimental validation or generate a new, whatever genomic data. So what previously in the homework, when we have a data, we told you, okay, you do X, Y, Z, that way you will get this result. It's like when you try to learn cooking from just a menu. They say, okay, 3 cups of this, you know, whatever of that, sugar and butter, and you can, oh, great, you can make a cookie. But really, next time, if you're not given this, it's like, oh, here, you have all the supplies. Can you make me a fancy cake? We want you to be able to at least kind of assimilate what we have learned so far. Okay, let's look at an example. These are the long answer questions from last year's final exam. Okay, so in a tumor profiling study, your collaborator conducted RNA-seq of 50 breast tumors, and they're matched nearby, like adjacent to normal tissues. And they also have some basic clinical information, like patient age, tumor stage, you know, survival information. There's no treatment, I guess, or maybe we don't do survival. Let's just say whether the patient recurred. Say these are stage 1 tumors, or no, sorry, different stage, say 1, 2, 3 tumors, and they just didn't have money to do chemotherapy. Let's just say this. And then we just ask, okay, some patient get cured right away, either the event is not death, maybe say the event is coming back for a recurrence. So now the preliminary analysis of the RNA-seq found that for many cases, the sample from the same patient are clustered together instead of cancers versus normal into 2 clusters. Just say this collaborator came to you and say, I used to collaborate with that group. They are trash. They don't know what's going on. We're getting this result. I'm sure it's wrong. Can you help me now? Describe how you would like to analyze this data. Just let's start from scratch. Starting from FASTQ. Yeah, after initial QC and normalization, are there potential downstream analysis you could do? You know, really now initially they came to you to say, okay, this group doesn't even know how to do batchy fat removal, but they say, okay, you're much better. Why don't we just publish this together? That's all the data we're gonna generate. What are you gonna do? From FASTQ. Sure, maybe you should do FASTQC first. Yeah, and you align it with, or you can do STAR. What else can you try potentially? You can try Simon as well. Sure. And then what do you do next after you have aligned all the samples? Sure. Yeah, but supposedly you run Simon, you will have TPM expression level for everything already. You might want to check the RNA quality in case these tumor samples have the degradation, right? You run like a RC-QC stuff, right? For QC your RNA, like make sure the coverage on the gene is okay. There's no degradation towards the 3', sorry, like a 5' get degraded, 3' are there. Other things, now you have the expression matrix.
Check the batch effect.
So now, how do you check batch effect? you do clustering, right? You can run PCA, you can run hierarchical clustering, you can do k-means or whatever, right? You can just do whatever. You can even do t-SNE, but that's too much. I think you can do just hierarchical clustering of the 100 samples. And indeed, you see that there's like 2 batches. Maybe they knew, maybe they don't. But you know, and the cancer normal in this batch is together. The cancer normal, maybe it's like, then you ask your collaborator, where did you get these tumor samples? Or when are they processed? Maybe you'll find out, oh, one is processed in May, the other was processed in August or something. Or maybe these batch were coming from hospital A, those batch are coming from hospital B. And so you run comebacks to normalize. After that, what do you do?
Just like a downstream.
Yeah, cluster again, make sure you have pretty much removed the batch effect, and bam, great. Now they are cancer versus normal, two classes. Or let's say when you cluster, you might have found that your normal is one group, but your tumor are 2 different batches. What happens there? So supposedly, in this experiment, they always process the cancer normal in the same batch. But if after that, the normal is actually pretty well clustered, but the tumor in 2 batches, well, what do you think there?
Just run comment.
Actually, at that time, because the normal already clustered together, you would think that your batch effect has been removed. And the fact that you see 2 clusters, it could be that in this cancer type, there are 2 subtypes. Like breast cancer, there's like a like luminal cancer versus basal cancer, or HER2. You know, like in lung cancer, the bigger class is like a adenocarcinoma versus squamous lung carcinoma, right? So there could be 2 classes like this. And then what do you do next? Differential expression between cancer and normal, right? You can run, what do you do? Like DEC, right? You can also check the 2 subclass, the subtypes to say, you know, maybe the subtypes have differential expression as well. And you get the differential expression, make sure you get to FDR adjusted. What do you do next? You could do gene ontology. You should always, in this case, do gene set enrichment analysis, because they tell you what other data in the public might also have that. You say if you do lung cancer, maybe they will tell you, okay, this one is really adenocarcinoma because other people have done this before. They have generated the signature, and your gene are skewed in the same way as those, right? Yeah, so gene set enrichment in the sense is that they kind of automatically give you other public data there. And you can ask what other, you know, like whether you see some strong signatures, some pathways or other differential expression showing similar trend as what you are seeing. What else do you do? Yeah, you want to see maybe in the 2 subclasses, or even you could do either, if it's only 1 cluster, you can ask, you know, what you need to run a survival analysis first. Say, we assume, well, we can check in the tumor stage, maybe patient age. You can check some of these differentially expressed genes, whether they influence your survival, right? Or you can just look at the 2 subtypes. Very first, to look at whether the 2 subtypes already have differential survival or kind of recurrence in this case, by adjusting the patient age and tumor stage. Yeah, I think that's kind of the starting point. If you want to do even fancier things, you can ask, oh, maybe immune infiltration in the early tumor can suggest whether the patient will have recurrence and other things, right? That's kind of a even higher level analysis, right? We'll give you these slides out. You can go back and check yourself, okay? Oh, I didn't mention since gene set enrichment, but yeah, you should totally do that. Oh yeah, I did mention cybersoil timer to look at immune infiltration, but you can guess there are some these basic ideas, okay? So that's one. The next one is in K562 cell, this is like a cell line. The ENCODE consortium has generated a whole bunch of data, histomark, transcription factor, you know, RNA-seq, DNA methylation, chromatin accessibility, and these have all passed QC. Don't worry, these are all done, assuming that you have like expression index or PCOS or whatever, all done. So recently, a group discovered a brand new histomark called H2XYZ. I mean, it's just like crazy thing, right? It's just like a new mark. Because there is a histone, like a histone variant called H2AX. But yeah, just like H2XYZ. And because of this, this collaborator has generated the first H2XYZ ChIP-seq data in K562. And they didn't do any perturbation or anything, just ChIP-seq. Can you help this collaborator conduct the initial QC of these data and try to understand a little better what this histone mark might be doing, or what's interesting about this mark? Okay, so you start from FASTQ. You have an input, you have a ChIP-seq. What do you do? Yeah, so you probably first need to do a FastQC. You read, what do you do to map? You use PWA to match, because this is DNA sequence now, right? Then you use max to call peaks. You might want to check the quality of the data, because very often the first time they do something, it may or may not work, right? So what are the ways you can check QC?
Check to see like what percentage of reads we call on peaks.
Yeah, like a FRIP score. What other QCs can you do? So if it has a very high FRIP score, it's a good. But even if it has a low FRIP score, it may or may not mean it's bad. It's just maybe because remember, every mark and every TF has its own characteristics, right? Can you look at conservation? Evolutionary conservation of the binding or the mark location? Yeah, if it's conserved, that's great. If it's not, well, maybe it's still okay. We might want to also look at its peak locations in the genome. So we have an algorithm for that. You have looked at that on the system. They say, okay, what percentage bind to promoters? What percentage bind to exons, introns, intergenic regions, right? You can look at its overlap with DNA's hypersensitivity to look at whether it's kind of in the enhancer regions or regulatory regions or not. But what other things can you do? Like a FastQC? Oh, that's past, yeah, I think, yeah, that's kind of, I think in the fast, no, that's not in fast. Yeah, mapability stuff, yes. And make sure they don't have too many repeat the same sequence over and over again, right? That's like a PCR bias. What are the things? That's your QC step.
You check to see if the peaks that you're finding like match any other.
Yeah, yeah, perfect. You run system to extract, you feed the peaks there and say, hey, what other data look like my factor? It's like, okay, well, this seems to be related to some other histone marks or related to some transcription factors. And remember, in this study, we do have I think all of these ENCODE data are already in Systrom anyways, but they also give you gene expression. Think about homework 5, the part you really hated. Can you see whether, so you can run something like a Systrom Go to assign the target for this binding. Initially, you don't even need to look at the expression, just because a Systrom Go works if you don't feed it expression. If you just give it binding, it will tell you some putative targets, right? And you can see whether the target that's assigned are transcribed genes or not transcribed genes. That can probably give you some idea whether this thing is gonna be in the active regions or the inactive regions of the chromatin, right? So something like that. And yeah, so you can, like FastQC, BWA, this is like QC, call peaks, QC, search for similar things, like find a target and look at whether the target genes are active or silent, right, as a start. Okay, so a few other scenarios. I'll just go fast. So supposedly we have 12 cancer cell lines of some cancer, the same cancer type, and 6 of these cell lines have a mutation on one gene, and 6 other cell lines have do not have mutation on this gene. They have some other mutations. And so your collaborator did a CRISPR screen to look at which gene influenced the cell line growth. They also have RNA-seq and ATAC-seq for the same 12 cell lines. This, I think, if you analyze it well, it can be published in Nature. What do you do? Yeah, so I would say the first step, I think for a lot of these cases, you need to make sure you do the standard QC and process data, right? There, you can use Magic to do the QC for the whatever the CRISPR screen. You can do your standard RNA-seq QC. You can use Max or whatever thing you do your ATAC-seq QC, right? All of these you can do. So the first step is to make sure each profile type actually worked. The second one is to probably do some comparisons, right? You compare your wild type, the 6 cell lines with the mutant 6 cell lines to say, are there some differential essentiality on the genes between the mutant versus the wild type? Do they have differential gene expression between the wild type and mutant? Do they have differential ATAC-seq peaks, right? You do the comparisons. And the third, it will be, like, oh, if these are different on these genes, do they belong to some pathways? Are they related to some cancer, drug response? For those genes that are differential or the ATAC-seq peaks that are differential, can we, so in some sense, for a lot of these data, I think a very good way to think about this, I first want to make sure the data is working. And then you want to make some comparisons to look at differences. And third, to ask, are these differences interesting, right? So those differential ATAC-seq peaks, are they correlated? Are they related to the differential gene expression? Are those ATAC-seq peaks bound by some important transcription factors? Is that transcription factor itself differentially expressed? Whether that transcription factor itself is influencing the downstream target genes or clinical relevance, right? And the third step is you pull other data types in here to help you understand the data better, okay? And next one is you have 10 patients treated with immunotherapy. There are 5 responders and 5 non-responders. And your collaborator is very rich. They did single-cell RNA-seq study. And so these are, say, melanomas or say, head and neck cancer, so they can take the tumor easily. They can get a pre-treatment tumor and a post-treatment tumor. So this is actually a lot, 10 patients, before after drug treatment, so 20 samples. Each of them is a single cell RNA-seq, whole lane of sequencing, a lot of data, right? So what do you do? Very first step, QC and process. What do you do? Yeah, so you use Surette for QC, mitochondrial reads, very low read number, very high read number, high read number could be doublet, low read, and so you use the standard thing to kind of get you the matrix, right? So make sure you have good readout. So that's the very first thing. Yeah, so you do QC, you do process, you can, and the next step will be the comparison, right? So what do you want to compare? For example, like before and after treatment for the same guy, are there differences? And between the 5 responders and the 5 responders, are there differences, right? So what are we talking about when we say differences? You can ask, okay, do they have, because these are single cell of everything, maybe the responders has more T cells, non-responders have more macrophage, something like that, right? So you can ask whether their immune cell infiltration is different. And, or maybe the drug can make the immune cell infiltration different based on the single cell. You can just look at T-SNE, oh, there are more things on here. You can also look at the differential expression between the, say, yes, these are CD8 T cells, but are the responders and non-responders showing differential expression, right? And also maybe after drug treatment, the same CD8 T cell become dysfunctional or become activated. So you can look at the differential expression between those different clusters or different patients, right? And then if there is something that really seems, in 5 patients, you might be able to see a little bit of something, but probably won't give you as statistically significant difference. You will see some trend, but you want to really see whether this is relevant. So now you check many other cohorts that don't have single cell data. You say, oh, based on this, I have an expression signature already. For this signature, I can run some other TCGA or some other public cohort that did just bulk RNA-seq, right? And I ask, when they have this signature or not have this signature, do they also show difference in survival, in response to other drugs, and so on? Right? So you can see a lot of these cases, when you have a data, you first do the QC, then you look at the differences, whatever they are interested in. And the third is you try to use other public data to help you substantiate, to prune the hypothesis. Which one, maybe not. Which one does have support from public data? That's the general approach you guys want to use when you think about a computational problem. Okay, so we go back to the very first. All biology is becoming computational, much the same way it has become molecular. I mean, I think in the 80s, there are people who refuse to use molecular biology. Boy, what a big mistake. And so if they don't, if they continue to look down on genomics and computational biology, they will be behind. But also, if they look at genomics, but not look at computational biology, this will be low input, meaning you don't think ahead of time, high throughput, meaning you generate a lot of data, and there's no result. There's just data. But so hopefully you will take what you learn in this course and do something great in computational biology. Okay, thank you.
Thank you.
Sure.